Welcome back to my YouTube channel. In this video I will be explaining radial immunodiffusion and this double immunodiffusion. First I will explain this radial immunodiffusion. It's also called as Mancini method. Why immunodiffusion? Because it is an immunological technique which involves antibody and antigen diffusion then form a precipitation, and that is why it's called immunodiffusion. For example, let's say this is the zone of antibody axis, this is the zone of antigen axis, and when the concentration becomes equal and zone of equivalence takes place and form the precipitation. Same is happening in this technique. For example, let's say this is an agar plate. Let's take this agar plate and distribute uniformly these antibodies on the gel. Then after create a well in between, or we can create many wells, and then after add the antigen in this well or a sample we can add to check the concentration of unknown sample. This way then this antigen will start diffusion radially. That's why it's called the radial immunodiffusion and form a precipitation ring. By this precipitation ring we can know the antigen concentration of an unknown sample. For example, these are different samples and these are the size of these precipitation rings. By this precipitation ring we can know the concentration of the antigen by plotting the graph between precipitation ring and the antigen concentration. More the precipitation ring, high is the concentration of the antigen. After that, double immunodiffusion. It's also called as Osterloni method, whatever you pronounce. It is the method which is used to visit for identification of antigens. Why it's called double immunodiffusion? Because antibody and antigen will diffuse at the same time. In radial immunodiffusion only the antigen is diffusing, because we first separate the antibodies uniformly on the agar plate. Here in the agar plate we add this part we add antibodies, and in this part we add antigens, sorry. Then these start the diffusing each other at the same time, then form a precipitation here. For example, we create 3 wells. Instead of these parts we create 3 wells, like antibody well, antigen, 2 antigen wells. This way this is called the double immunodiffusion, because these antibody and antigen will diffuse at the same times, not like the radial immunodiffusion in which we first separate the antibodies on the gel, then after we create a well and add the antigen. Then in this we create the well, then add the antigen and antibodies in these wells. Then after these antigen and antibodies will diffuse each other and form precipitation lines. For example, how to see the from these lines see the whether the antigens are identical or not. For example, let's say these are the 3 wells in the agar plate. For example, these are the 2 antigens and the precipitation line will be this shape. Then these 2 antigens are identical, and then after there are, if the precipitation line will be this like this shape, then 2 antigens are not identical, and if the shape is this way, then these antigens are partially identical. Identical means that 2 antigens having the same epitopes. For example, these are the 2 antigens, they must have same epitopes on the surface of the antigen, which will bind with the antibody and form the precipitation or bind with the antigen antibody reaction. Then after, this is the non-identical. That means that epitopes must be different from each other, and this is the partial identical. Some epitopes may be and same in the antigen. This was all about the radial immunodiffusion and double immunodiffusion. Thank you for watching.